Welcome to our global celebration of fungi. Without more ado, let me welcome all our speakers, and indeed particularly at the moment our first speaker, who is Nailoke Kadhila from the University of Namibia. Nailoke is an assistant professor there, and she has wide-ranging interests, particularly focusing obviously on mycology, but mycology, microbiology, molecular biology, and biotechnology. And one of her current interests is particularly on indigenous mushrooms and plants, and particularly those of medical significance. So, Nailoke, let me hand over to you to share your screen and to welcome you and to ask you if you'd like to give your presentation. Welcome, Nailoke.
Thank you so much. Thank you, everyone, for coming. I'm trying to share my screen now. Are you able to view my screen?
Yes, I can see that. Perfect.
Okay, I am Nailoke Kadhila. I am speaking to you from Namibia, or let's say Africa, and sitting in Namibia currently, and just want to show you where Namibia is with a pen straight zooming out my country, as well as some information about Namibia. I'm sitting currently at the University of Namibia. It's a small university. We are also a young university. We are just 30 years old. And we have about 12 campuses across the country. And within those 12 campuses, this year we have that number of students enrolled across all campuses. I teach and usually stay at main campus. And what is zoomed there, it's where I do mostly my research at the Zero Emission Research Initiative within the main campus of the university. I am going to talk to you about the African mushrooms, their cultural and socioeconomic significance toward the continent's sustainable development. I don't know what's going on here. So the African people have been using the mushrooms within their continent. We harvest the mushrooms, and our ethnomycological knowledge of identifying this type of mushrooms is passed on orally through our generations, and mostly the elders are the people who knows how to identify the mushrooms, and the young ones learn, and as they also get old, they get to know their mushrooms, be it for food collection or for treating their animals as well as themselves. Our mushrooms biodiversity attracts tourists interested in mycology, as well as those who just love to explore the nature. And this type of ecotourism activities also help the locals generate revenues and focus on other things where they can use the money for. In regions where certain mushroom species are in higher demand, most people engage in commercial, this is harvesting the mushrooms from the wild and sell them to generate income. And this is evidence if you move across African countries during rainy seasons, you will be seeing some people along the road selling mushrooms. This is also happening in my country. And the most mushrooms that are sold along the road in Namibia are the Temtomyces species, as well as some truffles. So these activities also nature conservation efforts to protect the habitat as well as associated ecosystem where this type of mushrooms are growing. In Africa, they also use mushrooms for ritual and spiritual uses. And These are mostly reported from Nigeria, Egypt, and other countries. I haven't seen any literature on the Namibian mushrooms on rituals yet. And some of these countries incorporate their mushrooms into spiritual rituals practices. An example I can give is the traditional worships using of these hallucinogenic mushrooms just for them to be able to do their activities. These also include ceremonies or rituals to connect with the ancestors, the spirit of wherever they found themselves, as well as sometimes to acquire the divine powers. They also use it in purifying themselves or the spaces where they are staying, as well as removal of negative energy or spiritual impurities in their dwellings. I continue with the rituals. Some communities also look and see mushrooms as the bridge between the physical and spiritual realms. So they use it as a connection or the spiritual strength to connect them to their spirit. Some cultures also use mushrooms as symbols of metaphors in mythology, in storytelling, as well as in proverbs. They also can represent concepts such as growth or transformation, or those which are looked as hidden potential of certain people. This is an example of mycologies that is used in different African countries. This is, I can't really see my titles, I can't see them. I do not know why. Just see. The table is just showing some species of mushrooms and what they are used for in medicinal aspects. As you can see, most of this research is done in Nigeria, Tanzania, in Uganda, as well as in Burkina Faso. I am also going to show what is done here in Namibia, because this table does not include my country, and we have also done some of medicinal ethnomycological aspects here in Namibia. So we have some of these mushrooms that are used in traditional medicine. We have the Themitomyces species, and on this one, I specifically zoom into the Themitomyces chimperi. This is just a recent study which we concluded last year, but it was only focused on one region, the Zambezi region, but I'm going to complement it with what I have done also in other 2 regions, in fact 3 regions of northern Namibia. So Themitomyces is used to treat sexual transmitted diseases. It is also used for heart diseases. It can be used for severe diarrhea. Some reports in the community are saying that they use it for cancer-related diseases, as well as kidneys, and in the prevention of miscarriage, as well as healing of the umbilical cord for the new babies when they are born. So that is on chimperi. And in other regions, I have also found out that this specific Themitomyces mushroom is used for ringworms. It's mixed with oil and applied on the heads of children, especially the boys who are suffering from ringworms. We have Ganoderma species in Namibia, of which we have actually got about, I think, 7 species of Ganoderma. And mostly when we were doing our survey, we firstly talked about the Ganoderma lucidum, and people are using it in the relief of stress or to treat shock, and they are also using it in the treatment of their cattle when they are suffering from lung diseases. They use it in a form of solution, which is drunk by the person who is suffering. Especially, they have described that when they hear somebody is in shock, for example, they receive bad news, somebody passed away. So they prepare this mushroom, they put it in water, and they let it sit for some few minutes, and the person suffering from the shock is given the concoction, and they drink it to just calm them down. Same thing is used for traumatic species, especially T. vesicola, which is also used in the treatment of shock, trauma, as well as stress. Same thing, it's also used in the liquid form. And then we have the puffballs. This is one of the interesting one. In some countries, it is eaten, but here I haven't seen any of us eating it. It is said to be used for the treatment of the wound, the wounds, as well as in contraceptive. This is, I did not get it from Namibia, but it is from a paper, and it says that the Yoruba women in Nigeria use it for contraceptive purposes. This is just a table taken from one of my first studies when I was, I think, doing my master's. And this is just having some of the type of mushrooms that are used in two regions, namely Oshana and Ohangwena, that are in the northern of my country. And it's got all those species. I could maybe talk out of Tephasia pefili, which is now referred to as Krahari tuber pefili mushrooms, which are also most abundant in my country. And these are used for eye treatment, and says that it is crushed and mixed with water, and that solution is then dropped into somebody's eyes for eye treatment. I can also hopefully have talked of the hexagon, which falls under the other mushrooms I discussed. And then we have the Amanita species. This is very special. It's a toxic mushroom, and it is used by the San people when they go hunting, and they use it to apply to the arrow so that when they shoot or they aim at the animal for hunting purposes, the animal gets to suffer from the poison and slowly die, and then they can use it for meat. This is just some of the pictures for the mushrooms I've been talking about. We have the termitomyces right on the far left of my hand, and the truffles, as well as some species of Ganoderma. We have termites here, and some puffballs, as well as another type of termitomyces. The recommendations I'm giving from what I've been gathering is that we need to continue the scientific research on the African mushrooms so that it contributes to the intensified knowledge and of identification, domestication, and the cultivation techniques of these mushrooms, as we can all know that we are losing diversity of most of everything, and mushrooms are not an exception. So we need to domesticate and see if we are able to cultivate this type of mushrooms. We also need to continue on the indigenous mushroom studies for the nutritional compositions, as well as their medicinal properties, which will improve or which will provide the insight for the value addition and commercialization of the products to be made from this type of mushrooms. We also need to do the detailed biochemical and microbiological research on these African mushrooms so that we will be able to get more from these valuable resources for the continent to flourish. This is just a citation of what research can be done. We can continue identifying our indigenous mushrooms in the continent. We can do the domestication of which or those which are possible, be it the edible ones or the medicinal ones, or even those that are regarded as unedible for the purpose of research and perhaps drug discovery from them. We also need to promote the cultivation for the local that are domesticated, as well as the exotic mushrooms that we get from around the world. I think it's the same title. We can work on our medicinal mushroom. This is a case of our own university. We have gone into the field, got some of those Ganoderma mushrooms, domesticated them, and we then went further to process them into dietary supplements, which we are now trying to commercialize and put in the market for the people to use. Still is just on the product, that is just show that that's what we did. We grow them in the lab. We then dry them, mill them, and capsule them and pack them as 100% dietary supplements for the Ganoderma mushrooms. We can also work on food, edible food. I mean edible mushrooms, which can be made into either porridge or cakes or biscuits or whatever it is. In this case, we use our most indigenous mushrooms that we are still unable to cultivate. We just process and add value by making 100% soup, or you can see the 100% Omajova mushroom soup. Omajova is the Temitomyces. We also make the truffle, and these ones we only do them when they are in abundance in the region, especially if you've got enough rain. If we do not get enough rain, we also do not get people selling this along the road or ourselves going to harvest them. So we do trials, and you can see last below insert of those are meatballs. I had a naughty student who just wanted to do something and fed everybody, of which it wasn't a bad idea. So we went on and we tried to add value by taking the normal beef meat, and we added some percentage of oyster mushroom flour, as well as the truffles and the Temitomyces, and we brought it on campus for everybody to test and rate which mushroom was best, or which meatballs were good with whatever flavor of the mushroom added. In conclusion, I would just like to say that whatever I have said here, the significance of use of these mushrooms may vary greatly depending on the culture, religion, as well as the historical context of where you find yourself. And mushrooms may hold a prominent place in the tradition and practices of some communities, but their eminence may be less pronounced in others, and the perception and use of mushrooms can also change over time due to cultural shift, the environment factors, as well as globalization. This is also a fact that as we acquire the knowledge on mushroom identification and mushroom uses, nowadays there's a lot of migration to urban areas, and as we move, we also create kind of an erosion in the indigenous knowledge that is being passed on, and sometimes people would not even say anything about it, or they know until less is known. But we really need to hold on to that and get to pass it on to the younger generations. We need to create awareness on the diversity and importance of our mushrooms in the communities, especially where there's less mycology knowledge, and this is going to help in the fact that if we tell the people the good of the mushrooms, be it about food or medicinal uses, at least people will be able to know that they shouldn't just over-harvest or overuse, and they will also probably develop the desire of growing their own mushrooms to have them available throughout the year. I think I have also said this before, the determination of the bioactive mycochemical compounds that need to be done, as well as the value addition, which might be an effective tool in addressing the challenges faced by the continent, be it poverty, hunger, and diseases. The mushroom products, if they are made, might as well allow consumers to enjoy the mushrooms, especially if somebody finds fresh mushrooms not palatable, they can probably drink the soup or eat biscuits that are made with a certain mushroom or have a certain mushroom flavor. The main thing is that we also need to have a genetic conservation for mushrooms and support. We also need to support the ecological balance to preserve this specific mushroom diversity. In acknowledgement, I would like to thank Prof. Boddy and Sally for keeping us on our feet, at least to be ready and to make sure that we come by. And I would also like to thank you, the listeners. Thank you so much for listening to me. And yeah, those are my citations, and thank you so much.
Thank you too, Nailoke. That was very interesting. We've had some wonderful talks today already. I thought it was particularly interesting how peoples in your country have been brought up on knowledge of mushrooms, and yet in other African countries they absolutely are not. And then it's the same in Europe and the same in Asia. So in Britain we're sort of mycophobes, and yet elsewhere in continental Europe, the people are very, very interested. And I know it's exactly the same in Asia, in very close proximity. Anyway, I shouldn't be talking about this now. We will come back to that, I'm sure, in the discussion. But thank you so much. Absolutely fascinating. My task now is to introduce our next speaker, and I very warmly welcome Bethan Manley. Bethan did her PhD at Cambridge, a postdoc with the Gurdon Trust, and is now working for SPUN. And for those of you who don't know about SPUN, I'm sure Bethan will be telling you very much more soon, but SPUN stands for the Society for the Protection of Underground Networks, and that's particularly mycorrhizal fungi. And Bethan's a computational biologist who's looking to understand the biology and genetics of global biodiversity. So very warm welcome, Bethan. Over to you.
Thank you, and thank you to the organisers for having me speak today. I'm very happy to be here on this UK Fungus Day international event. I'm just gonna. Okay, should be all good. So, as Lynn was saying, I'm Bethan. I am a geneticist and I specialise in symbiotic fungi. In particular, I work for SPUN, which is the Society for the Protection of Underground Networks, which is a non-profit science organisation that is based out of the US, but we have members and staff members all over the world. And at SPUN, we have this overlying concept, kind of like a motto, which is to protect the underground. So this is on a lot of our equipment that we use in the field, a lot of our outfits. If you're familiar with anything that SPUN does, this is a kind of an overarching theme. And what protecting the underground specifically means to us is protecting a particular group of fungi, and these are the mycorrhizal fungi that form amazing networks under the ground and form symbiotic beneficial associations with plant species. So it's really wonderful that fungi are getting a lot of attention. I'm sure many of you have noticed over the past few years that fungi are getting a lot of attention from the public and media. And this finally seems to be translating to some initiatives and protections to fungal species. But at SPUN, we would probably still argue that when it comes to understanding organisms and conserving them, there's still an above ground bias. So there's such a focus on the organisms that we can see and quantify easily, even when it comes to the fungal kingdom, that we may not be fully considering some really crucial actors in the functioning of a healthy planet when we overlook organisms that are beyond our sight. So we want to overturn this above ground bias at SPUN. But before I go into how we're approaching this, I just will talk a little bit about these fungi that we're so interested in that can be found mostly below the ground. So mycorrhizal fungi form symbiotic associations with plants, and the symbiosis looks something like this. This image is actually taken from a New York Times article that was written about SPUN almost 2 years ago now. It's a great article, so if you haven't had a chance to look at it, I highly recommend it. We made the front page, which was very exciting. And this image is a visualization of the mycorrhizal symbiosis, and it shows the mycorrhizal fungi in purple forming these out-of-sight networks in the soil. And what you can see here is that these fungi are really excellent foragers. They're actually better than the plant root systems themselves, because you can see that they have just so much more reach, they have so much more surface area. So the mycorrhizal networks actually help to explore the soil for water and for nutrients. And a lot of the time it's to an extent where the plants actually get a majority of their nutrients and water through their fungal partners and not actually through their own capacities to forage through their root systems. But the mycorrhizal fungi don't do all of this foraging and providing with nutrients for free. Fungi are like us, and they need to be able to eat the carbon-based products of photosynthesis that plants make. But these mycorrhizal fungi don't do it by farming plants like we do. They actually do it by living in very, very close contact with plant species and trading with them. So what you can see, what you can't see in this image actually, is that the mycorrhizal fungi are actually physically living inside the root systems of their plant hosts. They make these amazing intricate structures in which they swap their soil nutrients that they foraged for carbon from the plant. And these mycorrhizal networks really underpin terrestrial life on Earth, which sounds like a big claim, but really the organisms you see in a natural environment, so the trees, the invertebrates, the mammals, they're all supported by the presence of an underground network of mycorrhizal fungi. And these fungi sort of face a thankless task, because they, despite their importance in ecosystems, and despite the fact that they've been playing this really critical role for over 1 billion years, they're hidden from our sight in the soil, and it's difficult for us to see and appreciate their contributions. So you should be seeing a flow here. It looks like it's working. We do have some ways of seeing these mycorrhizal networks, and that's what I'm showing here, which is a short video of mycorrhizal hyphae. And what you're looking at is really a flow of information and resources. So it's a living network in the soil. It's dynamic, it's always moving. And this has been imaged thanks to a collaboration between AMOLF, which is the Biophysics Institute in Amsterdam, and the lab of Toby Kears, who is one of the founders of SPUN. And this image is not sped up. It's an incredibly complex and dynamic system, and it's extensive. So we know these networks can actually make up around 50% of the living biomass of soils where they're present, and there are just kilometers of them under our feet all of the time. But we don't fully still understand the extent of them and what they're doing. So a little bit more about the background of these organisms. So there are two main types. The first is the ectomycorrhizal fungi, and these include really well-known fruiting body-forming fungi like Amanita muscaria, which is our very charismatic fly agaric here in this picture. And although you can see these fungi for part of their life history, ectomycorrhizal fungi spend really a vast majority of time under the ground. So when you see a fruiting body, it's just the tip of the iceberg of the fungal form that is below the ground. These ectomycorrhizal fungi tend to form symbioses with tree species. They're very, very common in forest ecosystems, like I'm showing here. And the other group of mycorrhizal fungi are the arbuscular mycorrhizal fungi, which I will have to say are my favorite, because they're the group that I've always specialized in and worked on. And these never form fruiting bodies or any above ground structures. So they live out their entire lives under the ground. And one of the most remarkable aspects of these particular mycorrhizal fungi is that they can form symbiotic partnerships with roughly 80% of all terrestrial plant species. So almost any plant species that you can think of, that you can see, will be able to accommodate these useful little fungi in their roots. And the concept of fungal networks in the context of mycorrhizal fungi is that these organisms, both our buscular and ectomycorrhizal fungi, are really living networks. So their forms, their body structures, they are the actual networks themselves. So they are physical networks. They're always foraging, they're always growing through the soil. However, of course, as is the case with so many organisms on Earth right now, the areas where these essential mycorrhizal networks exist are being severely degraded. We're losing so many biomes where these fungi exist, and with their loss, we're really losing crucial components of natural ecosystems. So not only would the loss of these mycorrhizal networks destabilize entire ecosystems, but these fungi are actually, they're also a major carbon sink. So we know that billions of tons of carbon flows annually from plants to fungal networks. And so when we measure things like deforestation and the impact that this has on climate change by adding carbon back to the atmosphere, we also really should be measuring and trying to quantify how much carbon is being released by the destruction of soil mycorrhizal networks, because for all we know, they could be storing absolutely vast amounts of carbon underground that really needs to be understood. So mycorrhizal fungi support healthy ecosystems like this behind the scenes. These fungi are underlying the functioning of systems. They're crucial for carbon and nutrient cycling of the planet, and yet it's very difficult, say, to measure how they are coping with the changes that we're seeing across the globe. We need to know their resilience against temperature and land use changes, and we need to be able to ensure that they can keep supporting our ecosystems. So for many organisms, we can see how resilient they are, or how they're coping with change, and how their populations are coping through distribution mapping, which can be carried out thanks to observational data. So I'm showing this map here. This is an example that shows the species richness of amphibians. And this has been created and hosted on biodiversitymapping.org, and it's a really useful tool for seeing where the highest concentrations of amphibian biodiversity are. So it provides a target for conservation priorities, because you can see where the highest concentrations of amphibian species richness is. It's the places in red there. And this has been produced based on observations of amphibian species. So people citing these organisms and recording them, which is really, really useful. And this observational approach has been incredibly useful for many organisms, so birds, mammals, amphibians. But for mycorrhizal fungi, we can't map their distributions like this. We can't understand where they're located across the globe just based on physical observations. So at SPUN, one of our major aims is to identify key hotspots of mycorrhizal biodiversity. We want to be able to do this type of thing. We want to identify which ecosystems, which regions, which soils contain incredible mycorrhizal biodiversity, and hotspots where there are many species. And so this is a little bit about how we're doing it. We are trying to identify these hotspots that we can't easily see. We have to search for our mycorrhizal networks in the soil where they actually live. So scientists collect soil samples from regions of interest, they send them to a lab, and they sequence their DNA. And the DNA sequences then tell us which mycorrhizal groups and species are in the different locations that we've tested and taken soil samples from. And this method of collecting soil samples and sequencing DNA to see which mycorrhizal fungi are present has been done by the scientific community for many, many years now. So there is actually currently quite a lot of existing information and data on which mycorrhizal fungi are present at the locations that have already been sampled. And each one of these sampling points is really a snapshot into which fungi are present and how well they're doing. But taking these snapshots is not going to give us all of the information we need, because if we wanted to properly monitor organisms on a global scale to an extent as fine as what you can achieve through observational data, we would really need to be constantly punching holes in the ground all over the planet, which we don't really want to do. But what we can do is use DNA sequencing data that has already been collected across the world, and we can make machine learning predictions that extrapolate what we know about the areas where we have data out to the areas where we don't have data. So the data scientists at SPUN have a machine learning model that does just this. And this model is able to take lots and lots of data inputs to then estimate the mycorrhizal diversity that may be present all over the world. And this can be accomplished because we have many, many layers of different data. So we have the sequencing data, which you can see in the first picture. This tells us which mycorrhizal fungi are where. And then in the second picture, this is illustrating other layers of data that we have. So environmental data, so vegetation, climate, altitude, land use. And what we can do is combine all of this data to say, okay, based on the fact that these ecosystems at this altitude have this number of species, what can we extrapolate to estimate mycorrhizal biodiversity in other regions with, say, similar vegetation types and climate? How can we get a truly global prediction of mycorrhizal richness? And our maps look like this. So we produced two separate maps for the two main functional types of mycorrhizal fungi, ectomycorrhizal and arbuscular. And here I'm showing the prediction map of ectomycorrhizal fungi, and you can see the model predicts really high diversity of species in the northern latitudes, which is where we know we have these boreal and temperate forests that are actually the preferred hosts of ectomycorrhizal fungi. And so these hotspots, these areas where we have a lot of species or a lot of sequences, different types, are in yellow here. And this is the arbuscular mycorrhizal map. So it's a very different distribution. You may notice that's because these are very, very different functional types of mycorrhizal fungi. We know this type of mycorrhizal fungi are particularly prevalent in tropical forests, particularly in the areas of the world where there's a really high turnover in soil nutrients, as there is in much of the tropics. And these maps are an incredibly useful tool because as useful as they are, it's also important to emphasize here that these prediction maps are just that, so they're predictions. And a lot of work has gone into making sure that they are as accurate as possible. So we are constantly updating our machine learning models. This is an iterative process. So we're feeding them with more and more data as it becomes available, and more people are producing studies on mycorrhizal fungi. And we're also assessing the accuracy of our predictions using a method called ground truthing, which is where we take samples from regions or biomes where we have very few samples, and we test how accurate the model is able to predict in these areas based off a lower amount of samples than we would prefer. And one of the things that can really make biodiversity prediction maps imperfect or inaccurate is sampling bias. So this is what this image is really demonstrating. It's from a recent publication, and it shows the biases that exist in globally available biodiversity data sets. So just any biodiversity data set that's been collected on any organism. In black, we have places or locations on a map where there are 1 to 50 samples, and the yellow to red scale shows areas where we have over 50 records of collected biodiversity data. So you can see that there's really concentrations where tons of work has been done collecting biodiversity data, observations, understanding the biodiversity of these regions. But you can see there are huge areas of the world, the spaces between the black data points, where very few data sets that have been collected to study biodiversity of any type. So this is really a common issue across the whole of the biological and natural sciences. And these sampling biases mean we can't reliably model biodiversity in a way that accurately represents reality if our sample sets are skewed towards regions of the world with more scientific resources. So available data for ecologists and biologists to study biodiversity on a global scale actually really represents quite a small and biased proportion of the planet. And one of the approaches that we're taking to try and combat this issue of the sampling biases and how they affect our maps is in identifying the regions that have been undersampled from mycorrhizal fungi. So we're using information here on the known ecoregions of the planet. So We generate maps like this. They display the 850 unique ecoregions of the planet. These regions have been designated due to their unique biodiversity. So they have particular ecosystems, they have particular biodiversity that makes them unique to a particular region of the planet. And we're able to assess the numbers of samples that we have that are representatives of these ecoregions. We use maps like this to understand the ecoregions and therefore parts of the world where it might be valuable to generate more data on the mycorrhizal biodiversity that exists there. So the areas that are shown in dark grey, that's not just one dark grey ecoregion, it's just regions where we are not particularly interested in generating any more samples because we think sufficient samples have been collected in these regions for mycorrhizal fungi. So we have quite a lot of data from the ecoregions that are not shown, and those shown in colour could benefit from adding more data. And then what we do at SPUN is we put resources into getting more samples from these regions, because not only does incorporating more samples from more diverse regions of the globe help us to get more diverse data and hence a better model, it also means we can test the model and its predictions for parts of the world where we don't have many representative samples. And with the SPUN team have been able to go to some really wonderful places to collect samples for this aim. We've been to Chile, Lesotho, Italy, Ecuador recently, with more coming up. But it's actually not the team at SPUN that's doing most of this sampling, most of this work. We've formed a network that we're continually expanding of mycorrhizal researchers across the globe. And we want to support mycorrhizal researchers to carry out studies in their own countries and biomes of interest, since there are already just so many amazing scientists out there who are already studying mycorrhizal networks. We support our associated researchers with common protocols, collaborations, but our main method is by providing grant funding. So we have this network of scientists working together to produce data on the mycorrhizal fungal communities in the ecosystems that they study. And we know that there are serious funding and resource imbalances when it comes to science. So at SPUN, we don't want to perpetuate systems of science where researchers go sampling and produce studies that have little to no impact on the researchers and people that live there. So what I'm showing here is some of the locations where we've actually awarded research grants for one of our rounds of our Underground Explorers Grant Award. And if you'd like to find out more about the researchers that are these little representations on the map, please go to our website. They're all listed, and you can find out about their research and their projects that SPUN's working with them on. This work is really hugely important to us because we know that large-scale studies of biodiversity on a global scale don't always give us the data we need on how ecosystems are functioning and how resilient they are to change, because it's really local researchers who understand and know their own regions and ecosystems and biomes that generate the most useful data and enable us to fully understand what's going on. So with that, I'll just wrap up and give thanks to the SPUN team, because they have done all of this work that was described. There's many more of them now, actually, so I should really update the image, but this one is quite nice because we're all in our jumpsuits. So yes, thank you so much to the SPUN team, and thank you all for listening.
Thank you very much, Bethan. That was absolutely super. It's always a joy to hear about SPUN, and I'm sure that it's been really very revealing to other people. So now it's time to move on to the third presentation, and I think that's me replacing a speaker who was unable to attend, sadly. Let me see if I can share my screen. Practiced earlier and it worked, but then also it didn't work at one point. So let's hope that this is going to work now. Short answer seems perhaps to be no. Oh, okay, things are promising. Please let me know if you can't see that, Sally. So perhaps I should say who I am. I'm Lynne Boddy. I'm a fungal ecologist. I was an undergraduate at Exeter, a postgrad at Queen Mary, London, postdoc at Bath, and then for the last more than 40 years, I've been working at Cardiff University in South Wales. And my particular interest is in wood decay fungi, their ecology in all sorts of ways, including, I should say, networks, climate change, etc., etc. But I just quickly put together a talk here just to fit in for this half an hour slot, called Home is Where the Heart Rot Is, because this is one of my current huge interests. So throughout their lives, trees can produce and form a huge amount of habitat and niches for fungi, food for fungi and other organisms. But this changes throughout the lives of the trees. Trees can be long-lived, some can live even for 1,000 years, such as oak trees, unless we humans get in the way and chop them down or whatever. And they grow up and have very little decay, usually to start with, until they become well mature. And then some of the limbs become dysfunctional because perhaps of lack of light or whatever. And the centers of the tree trunks just begin to start to hollow. When they reach the ancient phase, the crown starts to retrench. And what that means is that the huge canopy that there might have been to start with starts to die back somewhat. And this makes available branches for colonization. And as time goes on, this happens more and more. The trunks become rather larger. So rather like, probably like most of us humans in middle age, and they start to shrink in height and to extend in girth. The center of the trees become more and more decayed until in the final late ancient stage, the trunks are indeed dominated by wood decay. And that's what I'm going to talk to you about. But first, let me just show you some actual photographs of these sort of things. So this is for oak trees growing up at early to mature stages, fully mature on the left-hand stage here. In the middle, you can see the crown just beginning to reduce. You can see this branch here, once the canopy extended here, but these branches have died and are available for colonization. And then in the mid to ancient phase, you can see the canopy used to be really extensive, but is now a shadow of its former self. And then in the late ancient stage, typified by this Queen Elizabeth I oak tree, this huge hollowing gone on. This tree has got a girth of getting on for 13 meters, but you can see that there's not very much of that tree trunk left. Most of it is hollowed out, but still the functional sapwood supports quite a lot of vegetation here. And it's not only oak trees, there are many trees which grow to this extent, given half a chance. The one on the left here is ash, and this is beech on the right. Now, those trees you've seen so far with the hollowing centers are really rather large, and this is often the case, but not always. Depends on tree species. So on the left here, we have an example, Elder Sambucus nigra. Now, this tree is, I suppose, perhaps just 20 centimeters across the trunk of this. It's a hedgerow tree, and yet you can see it's hollowed. It's an old tree, but it's not large. These trees don't reach great sizes. And then again, you can see at the extreme here on the right, we have a bonsai tree in a greenhouse. This is a very old tree, but it's not a large tree, it's a bonsai tree. And you can see there's the entrance to a large hollowed cavity inside. Now, the heart rot is extremely important for many reasons. It's important for nutrient cycling. And if you look into these hollows, you can often see material which is more like soil than wood. This picture on the right-hand side here is probably well over a meter up the tree. And you can see this looks like soil. These are roots that you can see here. It's a beech tree, and the roots have grown down from inside the tree, from higher up down into this, what is now soil-like substance. So it's decaying wood. With lots of roots in there, the fungi causing the heart rot, a rot in the wood, releasing the nutrients, and then the roots that have come from the tree can sequester those nutrients and use them to carry on growing. It's feeding on itself with the help of fungi. And there are a few examples of roots growing down into rot holes. Now, second reason why heart rot is important, obviously, is for fungi, for very many fungi indeed. And I just illustrated here 3 examples of threatened species. On the left here, we have the rare oak polypore, Bulgossoporus quercinus. This is threatened globally. Probably the world's stronghold is in Windsor Great Park, in the oak trees there. And then these two species, these Hericium species, are both threatened in Britain and in quite a lot of Europe, but equally well, they're common in other parts of the world, so in parts of Asia and also in North America. Heart rot is also important for animals. Worldwide, over 1,800 species of birds and mammals, and indeed also amphibians to a lesser extent, and reptiles, depend on these habitats, illustrated here by a woodpecker. And there's a woodpecker hole. And of course, it's not only the large animals that depend on this habitat, but also invertebrates too. Now, I don't have a figure for worldwide, but in the UK alone, over 1,700 species are dependent on this rotting type of habitat. And this is over 6% of the invertebrates in the country, and includes 15% of the rarest species, such as the violet click beetle, Limnoscus violaceus, which is found occasionally in beech and ash. And I've indicated a bit more the sort of habitats you find for invertebrates in trees. So I've just mentioned the hollows inside, but there are all sorts of other habitats and different types of hollows, sometimes filled with water, which attract different invertebrates. There are the fungi themselves, which form breeding grounds, feeding grounds for invertebrates, mycelium growing on parts of the wood, and of course, various features of the tree itself are important. And just a few examples of some of those invertebrates that I mentioned, ranging from things like pseudoscorpions to mites and spiders, collembola, various larvae, snails, rat-tailed maggots, and beetles. So let's stop and think a moment about how fungi actually get into the heart of the trees to perform decay, and when they can do it. So let's think a bit about the structure of trees and how they function. I mentioned a little bit about this in the discussion session this morning. So in many trees, the functional part of the wood, functional in terms of moving water from the roots to the shoots, is in the outside. In some trees, it's in more central regions too. Fungi can't grow in these outer regions because they're full with water. At least basidiomycetes can't grow very well, the wood decay fungi, because of this high water content. But in the central regions, eventually that stops conducting water, and eventually the gaseous regime improves, there's more oxygen in there, and fungi can colonize. How do they get there? Well, some can certainly get in from attached branches, whether they've got heartwood in them, or in some cases, whether they haven't. For example, the Fungusterium gaussipartum is a common branch decayer in oak trees, and it also can colonize heartwood and probably tracks down into central tissues and causes this sort of pipe rot that you can see here. Other ways of getting in are through large wounds exposed when limbs are broken off. You can see fungi fruiting here on some of these exposed heartwood regions, but don't necessarily think that these particular fungi that you can see here actually got in through this particular wound. The chances are they got in very much sooner from other places. It just so happens that in this case they're coming out of these particular wounds. Then, of course, they can be left behind in small bits of dead material. So twigs and branches which are colonized are engulfed as the tree grows outwards, because remember, the trunks grow outwards and can engulf the sort of dead and decaying bases of some of these twigs. You can see this here, and this is a classic example is the North American species Echinodontium tinctorium, which is left behind and then subsequently can colonize the wood and indeed fruit beneath the original twiggy branches. Now, actually, I've just mentioned that you tend not to get fungi being able to grow in functional sapwood because of the high water content. But there are many fungi present in that environment. You could call them endophytes if you want to, or we might say that they are latently present. And we've sampled standing trees, and these are data for beech trees. Now, there are not very many fungi or fungal propagules in there, presumably. It's not extensively colonized. I explained that fungi can't do this. So we're sort of looking a bit for needles in haystacks. And in that situation, you have to be very careful about your sampling. You mustn't get contaminants from the air or from the bark or whatever. So we've been very careful in our sampling process so that the drill samples are never in contact with anything other than the wood inside. They're not in contact with the bark, and they don't have spores landing on them. So the drill bits are encased in several layers of plastic. They've been sterilized, and samples are collected very, very carefully. And this revealed to us then, we're pretty confident that the fungi which we find in the sapwood, which we then extract the DNA, find out what we've got there. And we have a huge long list of species here of white rot decay fungi, brown rot decay fungi, soft rot fungi, which are mostly ascomycetes, and we're pretty confident they come from actually within that functional sapwood. And amongst these, there are heart rot decayers, so that's probably another way in. And then a sort of a final way is that they can arrive below ground. They can arrive when roots contact each other and maybe are grafted. This is how Heterobasidion and Newsham colonizes. They can arrive as rhizomorphs, such as rhizomorphs of Armillaria gallica that you can see here, or indeed as mycelial cords of other wood decay fungi. So what else do we know then about heart rot? Bearing in mind that very little work has been done. I was trying to get money to do this for a very long time. It's only about 7 or 8 years ago that we managed to get money to start to work on beech, and then more recently on oak, and then on ash. But what we do know is that there's a range of selectivity for fungi involved. So some are extremely selective. I've already mentioned the rare oak polypore, Bogossaporus quercinus. This is extremely selective for oak. The beefsteak fungus, Fistulina hepatica, also selective for oak, but also for sweet chestnut. Others are less selective. Sometimes they are found on a few unrelated tree genera. And I used to use Laetoporus sulphureus as an example for that, because we see Laetoporus, for example, on oak, quercus, and on yew, Taxus baccata, rather different trees, and indeed other tree species too. But of course, nowadays we found out that there are many species of Laetoporus which superficially all resemble each other as far as the fruit body is concerned, but indeed there are different species there. So I think there are 4 species in North America, and there are 14 or 15 or perhaps more around the world. And some of the heart rot fungi are widely occurring, for example, the armillarias, which occur on all sorts of different tree species. Now, if we consider the fungi present based on fruit bodies, again, we get a different story depending on trees. So, for example, if we look at oak, we find that there's a relatively small number of fungi reported to fruit on the trunks of oak trees. If we look at beech, on the other hand, there are rather more. And this sort of makes intuitive sense, because oak has heartwood, which is full of tannins and other polyphenols, which are inhibitory to enzyme function. They have very low pH, about 3.5. On the other hand, beeches, shall we say, in inverted commas, are much more friendly environment as far as fungi is concerned. So you can imagine over evolutionary time, relatively few fungi have evolved to cope with those horrible conditions in oak. But this is fruit body data. Let's see what actually is happening. And we've adopted several approaches. We've adopted a coring approach, so we can take cores out of standing trees, like you can see here. Here is a core, and then we can chop this up into tiny bits, 1 centimetre sections. We can plate these bits out onto malt agar, see what grows out. We can extract DNA directly to find out what fungi are present in those standing trees. And then also sometimes trees fall down or are taken down for various reasons, and then we can cut sections out and try and build up a picture of the 3-dimensional community within the wood. And when we slice wood, we see all sorts of patterns like this. So this top pattern here, these patterns actually are produced by the tree, probably in response to the tree gradually becoming aerated, and these are bands of thicker wood probably put down by the tree to maintain the integrity of the water conducting column around here. Other patterns that we can see are caused by fungi or other microbes. So here, this dark staining is called, well, I will call it wet wood, bacterial wet wood. It's wood in this case, which has become wettened, usually in beech, by ingress of water higher up the tree, and then bacteria have taken over. In some tree species, there's a pathological condition called bacterial wet wood. And then here, in these bottom 3 pictures, you can see these interaction zone lines. That's where fungi are interacting with each other. So this piece of territory here is occupied by probably one major decay fungus. Likewise, there's one major one in there. And then in other cases, there are lots of different individuals. They demarcated, their territory is demarcated by these structures all around them, these pseudosclerotial plates, a bit like how we used to defend our medieval cities, or perhaps put a wall around our homes and gardens. And we can get a sort of a picture of what's going on. So this is just one beech tree. Here's the tree, and you can see where the different sections have come from. And what surprised us immediately when we analysed this tree was that we got Pholiota sclerosa from much of the tree. There's a picture of it. And yet this isn't listed as something which you find on beech, perhaps because it has fleshy fruit bodies, which are not around for very long. But this occurred time and time again. Oh, and incidentally, perhaps I should say that this tree, before it was felled, there was no indication at all that there was any decay in that trunk, no indication visually from the outside. Here we've got just another example, and perhaps we'll just look at this basal section here. Oh, well, there you go. There's one we were expecting, Ganoderma australe. That fruits commonly on the outside of trees. Oh, the Pholiota was there again. And then there was Sterium hirsutum. I would have never guessed the Sterium hirsutum was a heart rot fungus. We normally find this on branches and small branch litter, etc., on the forest floor, don't we? And then here's another one which surprised me. Hyphiloma fasciculare in this basal region. The sulfur tuft fungus, we often see it on stumps and other material, but I never thought of it as going to be a heart rot fungus, but there it is. I'd always thought that it was on stumps when they're felled and the spores land, and indeed that's very often the way they colonize. But clearly it's also present as a heart rotter, as is this fungus, Anascomyces utopa spinosa. So there are 3 different ways in which we can find what fungi are present in the hearts of trees. We can do the coring method, the sectioning method, or indeed look for fruit bodies. And we've listed some of the main species involved here. But those which I've put these red rings around, you'll notice that those are not things listed as being present as fruit bodies, but they are important fungi actually as heart rotters. So just to tell you something which we already knew in truth, didn't we? Fruit bodies are not always a reliable indication of what's going on inside. Okay, let's turn our attention then to oak. So this is a slice through an oak trunk, and you can see this central region of heartwood, very dark coloured with its tannins. This is the sapwood, the functional part, and we can take chips out. Use the same sort of approaches with coring and slicing, although we haven't been able to get many trees which have fallen down. You'll notice that there aren't all these interaction zone lines in oak. And that's probably because it's largely decayed by brown rot fungi. So we've concentrated on coring, and this indicates the extent of fungi in the core. So on the left here, this is the outside of the tree. On the right, that will be the inside. And this represents where a fungus is found along the length of one of these cores, from the outside of the tree to the inside. The red colour represents Fistulina hepatica, you can see top right. Yellow is Laetiporus sulphureus, grey is Grifola frondosa, and this was something else that I can't remember what it was offhand. You can see how in these 9 or so sections, Fistulina was dominating, sometimes with Laetiporus in the middle. We made isolations and sequenced to find out what we've got. Now, if you look, this pie chart here represents the diversity of fungi based on the number of species there were. Only 9% of the species found in the heartwood of these oak trees were basidiomycetes. 90% were ascomycetes. So if you're keen on the ascomycetes, here's an indication of what was present. But remembering back to those previous to the previous slide, which I could just flick back to there, you can see that actually that doesn't give a fair impression of which fungi are dominant. Fistulina is clearly dominant in these. It was found throughout. If we look at the basidiomycetes, we find that in 27% of our cores we found Fistulina, 22% Laetiporus, but as I've said, Fistulina really is very often dominant. Sometimes we found both in quite a lot of the trunks which we looked at, even though the trees were usually probably over 220 to 250 years old. Quite often there were no decay fungi even then in the centre of those trees. Now, just before I finish, I'd like to turn to a problem that we have, and that is the generation gap. So although trees are often long-lived, as I said, perhaps the oaks live to maybe 800,000 years old, they do fall. Now, in Britain, we're very lucky in that we probably have more of these ancient trees than anywhere else in Europe put together. But nonetheless, these trees are aging and they're falling and they're disappearing. Now, as you know, there's lots of tree planting programs going on. But you can't plant a 200 or 300-year-old tree. And we have a generation gap. We still have quite a lot of these veterans and ancients. We have quite a lot of young trees, but not many in the age class between. So the worrying question is, what happens when our veterans and ancients have gone? There's nothing to fill the gap. And yet there's such important habitat for all sorts of organisms. So to try and solve that problem, people have started to try to veteranize trees, in other words, to make young trees have older characteristics. And one approach that's been adopted has been to try to make features similar to what we'd see in the veteran trees. So, for example, cutting holes in the trees, perhaps to simulate woodpecker activity, hit the bases of the trees with sledgehammers or something to simulate horse damage or large mammal damage, cut holes in to mimic lightning strikes, and other holes to mimic some of the structures that's seen, or to ring bark branches. And indeed, these activities are forming habitat which is being colonized by various organisms. But of course, it really, you have to be lucky to get the right fungi at the right time to develop exactly the habitat which we get in nature, and to get all of the different types of habitat which we see in the natural environment. And so we have adopted a slightly different approach, a more targeted approach, and that is to inoculate young trees, youngish trees, with heart rot fungi. And this is work that's been largely done by Matt Wainhouse when he was doing his PhD with me. And Matt isolated fungi that we know to be important heart rotters, grew them in culture, then put blocks of wood on the cultures that we got and colonized them for 3 or 4 months. In the case of beech, we're doing work on oak now, which is taking more like 6 months to colonize them. Also colonized sawdust for packing around the side, and also tried dowels. We then cut holes in trees and inoculated the fungus and packed around it, and then subsequently monitored to see what happens. And I'll just quickly tell you about that. One thing I ought to emphasize, that it's absolutely essential that you only use locally sourced strains of fungi. It's not appropriate to go and buy anything that you see on the internet and think, oh yes, this will be a good one to inoculate. Usually we don't know where these sources are from. Quite often they're from outside of the country where you're buying them. And it really is not appropriate to be importing fungi from other places in the world, which might then establish in the natural environment of your country. You have to source your strains locally, never from unknown sources. Also, I emphasize these points on the right, that you should never inoculate trees that have already got veteran characteristics, because we'd be interfering with the natural processes. Only inoculate the younger trees. And I should also emphasize that we're not trying to kill the trees, we're only trying to colonize the center of those trees. So we cut holes in, we inoculated the fungus. We actually inoculated in our main experiment with 4 species, a couple of Ganoderma foliota and a Hericium. And we alternated, we inoculated on several different years. And sometimes we cut the hole early and sometimes late, the day before we were inoculating. And so the first sort of monitoring you can do, obviously, is to look at fruit bodies, but we know the problems with that, of course. But here are fruit bodies appearing 2 or 3 years after inoculation. So here we've got a Coprinus fungus growing on the packing material. There's the ozonium state of Coprinus. There's the Coprinus again. We did not inoculate Coprinus. We did not inoculate this. We did not inoculate this. Yes, we inoculated this, a Ganoderma. We did not inoculate this, a Hypholoma. Yes, we inoculated various Hericiums. So from that, one might suggest mixed success, but let's try a better approach at sampling to see what is actually inside the tree. And we took drill samples from above and below the inoculation, and then extracted DNA to find what was there. These are indications of above the inoculation. This is below. A small blob means 1 of the 3 samples was successful. A larger blob means 2 of the tree sample was successful. And so you can see that very often we have had success. We never had success with Ganoderma pfeifferi. You'll also note that we found quite a lot of other species were colonizing as well as the thing which we had inoculated. And then for all of our trees, last year, some guys from Sweden and also a couple from Britain very kindly sampled all of our trees. When I say sampled, that's not really the right word. They did perform sonic tomography on them. That's sort of looking at what the state of the tree is like inside. So you insert all these probes around the tree, hook them up to the computer, and then you hit each probe in turn. And all of the other probes measure the time taken for the sound to travel through to another probe. So you hit that one and it measures the time to get to all of these other ones. Now, sound travels quickly through solid wood, much more slowly through decayed wood. And from this, you can build up a pattern of what's going on. And this is the computer screen. You can see on one of the days it was raining as the rain drops. And the darker red color indicates when there is decay. So there's a nice indication there of decay around our inoculum. Now, we would like to have been able to slice up some of these trees, but this is a long-term experiment. It's on a site which has got to be left standing forever. But Storm Eunice came along and blew down one tree, which hit one of ours. And here it was. So tree 90 was then fair game for slicing up to see what happened. So we sliced up tree 90. This was the point of inoculation there. And look at all the decay that it established. And indeed, there was evidence of activity almost 5 meters up the tree and also below the tree. And a year later, from the fallen samples that were on the ground, it was very well decayed. You could put your finger in. Now, admittedly, that decay could have occurred more rapidly once it was down, but we are certainly having success. So on that note, I would like to thank everybody, and there are a huge lot of people who were involved with this work. And thank you very much for your patience. And it's now time for us to move on to the fourth talk, the final talk in this session. So I'd now like to invite Kevin Newsham to talk. He's going to tell us about a continent which we haven't mentioned yet, and that is Antarctica, but also the Arctic, so the extreme polar regions of our planet. Kevin did his PhD actually in Cardiff a very long time ago. I won't say how long ago because that ages both of us. He's then worked he worked on pollution actually in those days, when sulfur dioxide was all the rage. Fortunately, we're not polluted so badly with sulfur dioxide nowadays. Also worked with mycorrhizal fungi subsequent to that, and then moved to work for British Antarctic Survey in these extreme environments. So Kevin, thank you very much for coming to talk to us again, and let me hand over to you.
Okay, thanks, Lynn. I'll just share my screen. Let's see if this works. Does that work?
Perfect.
And can you see the pointer?
I can see the pointer, yep.
That sounds good, great. Okay, so thanks everybody for coming along and listening. And thanks for Lynn for that nice introduction. So hang on just a second, let me see. Just move something on my screen. As Lynn said, I'm a mycologist. I've worked for BAS for the last 25 years or so. I've got around 30 slides describing BAS and its logistics. Then I'll tell you a little bit about what polar environments are like. And then I'll describe climate change in polar regions. And finally, I'll go through experiments in the Antarctic and the Arctic that are aimed at trying to get to how fungi will respond to climate change in these regions. I've got quite a lot of information from the Antarctic rather, since the experiments there ended about a decade ago, but I've still got ongoing experiments in the Arctic, so that's still a work in progress. The talk's quite general, so it's pitched at around 6 formal levels, so 16 to 18 years, so I hope most people can understand it. There's no what I call hard science in the talk at all. So if you're a research level mycologist, and I know there's at least a couple in the audience because I've seen their names, probably now is a good time to put kettle on. So BAS is a component of NERC, which is itself part of the UK Research and Innovation, or UKRI. It was established a long time ago in '43 in something called Operation Tabarin, which was a mission to gather net data from the South Atlantic for Allied shipping. Nowadays, we have over 450 staff, most of whom are based at this site in West Cambridge in England. We have 5 research stations, 5 aircraft, and a research ship, the David Attenborough, with a very well-known ROV called Boatface. These are some of the first people who went down to the Antarctic in '43. Just a little bit about the logistics of BAS. It operates an incredible every summer it transports hundreds of people down to the Antarctic and back again, so it's a remarkable operation. So most people travel south on a jetliner to a place called Punta Arenas in Chile, or via the Ascension Islands on an RAF flight to the Falkland Islands. This is Stanley, a new city. And from there, it's about a 5-hour hop across the Drake's Passage in this aircraft, which is a de Havilland -7. a super reliable aircraft with 4 turboprop engines, so I feel very safe when I'm on this plane. This is Rothera. It's on Adelaide Island, which is here. It's about halfway down the Western Antarctic Peninsula. It's jovially known as the banana belt, this region of Antarctica, because it's marginally warmer than other places in the Antarctic, although not a lot, as you can see from the photograph. There's about 100 people on this station during the height of summer. Rothera is quite an unusual place. There's no money, everything is on tick. There's no TV, and there's a pretty limited internet connection as well. There's the chef, so you don't have to cook yourself, which is great. But there's set times for meals, situation reports or sit reps, and then there's the base commander whose word essentially is final. The accommodation is very good. Nowadays, it's 2 people for a room. It used to be 4, so which was a nightmare if you were with a snorer. And then there's a very, very good research laboratory called the Bonner Lab at Rothera. So, the fairly obvious is Antarctica, since it's cold, but how cold? Well, these are the Leone Islands, close to Rothera. This is in Ryder Bay. These islands are about 10 kilometers southeast of Rothera. This is Leone Island itself, this pyramidal island here, and then this lagoon and Anchorage Island in front. And daily average air temperatures are about, well, a hot day during the summer will be about +5 degrees C. You can get warmer, but it's pretty rare. And then temperatures can drop to about -5 degrees C during the summer. The rest of the year, you're lucky if you're above freezing point, and it can drop to about -30. So it's cold, but it's not desperately cold. There's certainly life can take hold in these environments, and if you're carefully on Leone Island, you'll find plenty of plants and fungi, as you'll see in a second. Aridity is perhaps the biggest problem for life. It's not really the cold that's the issue, it's mostly the lack of water. So most of the water is in a solid form. It's not liquid, so therefore it's not biologically available. That makes things really tough for most forms of life. Wind is a big problem as well for plants, because you have extremely high winds. This is a field camp with the best standard bamboo pole stuck in the ground, and the angle at which it heaves over is an indication of how quick the wind is. This is an upside down aeroplane called a twin otter. I suspect somebody was not very popular in the morning when they discovered this. These harsh conditions strongly affect which plants survive in these environments. So mosses are the main plant form in Antarctica. There's also a very interesting plant form called a leafy liverwort that has fungal associations in it. I haven't got any slides on that today, but it's one of my great, it's been one of my great passions over the last few years. Woody plant species are entirely absent from the Antarctic, so there's no woody plants at all. So this is a diagram showing the main types of fungi in about 30 soils. This lack of woody plants strongly affects these fungal communities. So there's a very well-known mycologist in Estonia called Lehto Tedesuu. When he published his paper in Science in 2014 on the soil fungal communities on all of the continents, he found that about half of all fungal types were basidiomycetes, just like Lynne just described in her wood rotting talk. But because there are no woody plants in the Antarctica, the soil fungal communities are completely different. So they're mostly ascomycetes. So everything on this diagram here from about 1 o'clock going anti-clockwise all the way around to about 3 o'clock, these are all ascomycete taxa. whereas the basidiomycetes, which Leo found to be very frequent in temperate environments and tropical environments, these are much less frequent in soil. There are certain fungi as well, or certain taxa at the genus level that are very frequent. So this one in particular, Pseudogymnoascus, which used to be known as geomyces, is super frequent within Antarctic soils. And in fact, in this study, this was the only genus that colleagues and I recorded in all of the soils that were taken. Toadstools are really rare. They do occur though. This is probably a Galerina growing on Leone Island, the pyramid-shaped island that I showed a few slides ago. And then, as Bethan said, you can take DNA from soil, you can sequence it to find out which fungi are there. And so the taxa, the pseudomycetes that form toadstools, if you like, or sporocarps, as you go further into the Antarctic, so moving from 60 degrees of latitude down the Antarctic Peninsula, down to 72 degrees latitude, these fungi become less and less frequent in soil. On the flip side, lichens are really abundant. So there's a field of lichens on the South Shetland Islands taken by my colleague Elise Bjørsma, who's now in Copenhagen. And this shows the lichen Usnea growing along a beach somewhere in the South Shetlands. Lichens are symbiosis between fungi and algae, normally, sometimes cyanobacteria, but less frequently so. And there's about 400 Antarctic species of lichens. The algae in them can photosynthesize at temperatures as low as -20. That's about the same temperature as the inside of your freezer. So they're pretty hardy symbiosis. And they're what's known as poikilohydric, which means they can survive really long periods of drought in a dry, inactive state. But as soon as they get re-wetted by liquid water, then metabolism recovers very, very quickly, and they can resume normal business. And in the dry valleys, at least, the rate of growth of these organisms is something like 0.5 millimeters per century. Alan Green, who's worked in the dry valleys, shows a quite funny slide of a lichen growing on a rock that he visited something at the start of his career in the, I don't know, say the 1960s. And then he's a picture of the same lichen from 40 years later, and it's exactly the same. It hasn't changed at all. Now, I mentioned this fungus. This is Pseudogymnoascus. So this is growing on an agar medium on a petri dish. And this fungus is able to grow at about -35 degrees C. These people called Honeycuff and Sizova in this study, they grew this fungus at sub-zero temperatures and found it could grow up down to about -35. But they also say, we calculate that doubling time increases with further cooling, reaching 20 years at -18 degrees C and 62 years at -20 degrees C. Doubling time is the time taken for one cell to divide into two. So it may grow at -35, but it's very, very slow. Snow molds are perhaps one of the best examples of fungal adaptations to freezing. Moss beds in the Antarctic, also the Arctic as well, are really good places to find these fungi. You get these horseshoe-shaped infections in the mosses. Some of these fungi, such as Typhula eukaryotes, grow at less than 0 degrees C. See here, this graph shows temperature on this axis, and then growth rate up here, and at -5 degrees C, the fungus is growing. Again, it's quite slow, but it can actually grow. And the reason for this is that this fungus produces antifreeze proteins, which absorb onto the surface of ice crystals. So when ice crystals grow and they elongate, they punch through the cell walls of fungi and other animals as well, other organisms as well. Now that's what kills the cells, but these proteins, they adhere onto the surface of the expanding ice crystal and they stop it from growing, which is how the fungus survives. I'll say something now about climate change in polar regions. So the poles are warming more rapidly than anywhere else on Earth because of something called polar amplification. And this is caused by a loss of sea ice. And sea ice reflects about 85% of radiation back into space. So if you will, imagine sort of radiation coming in, and about 85% of that energy or radiation is lost into space. However, as ice is lost, more sea and land become exposed, and these dark surfaces reflect only about 7% of radiation back into space, so much, much less radiation. And the problem for polar regions is that this generates something called a positive feedback loop, whereby as the sea ice melts, or rather ice and land can melt as well, this leaves a darker surface which can absorb more heat, which causes temperatures to rise, which causes more ice to melt, and it's kind of this round and round, positive feedback effect. And that's the reason that Antarctica and the Arctic are warming quite so rapidly. Maritime Antarctica itself, so the peninsula region, has warmed at about 0.4 degrees C per decade over the last 70 years or so. So these are data from 3 stations along the peninsula. These are raw data. So I know that scientists sometimes get in trouble when they seem to be misrepresenting data or whatever. These are raw data taken from an online publicly accessible database called the Reader database, and they show over time on the x-axis in years changes in annual air temperature. And you can see, because there's natural variation, that there's a fair amount of, it's not a straight line up, but you can see if you look at the points that there is most definitely warming going on there. And then there's another 3 stations here on the other side, or rather the northern part of the peninsula. One thing I'm really interested in is how soil fungi will respond to climate change. And there's 2 main ways to get at this. One can use a latitudinal approach where you sample soils or whatever substrate from increasingly cold environments. So here, moving down this transit down the peninsula, this is also called a space for time approach. And there's also a manipulative approach using something called open top chambers, where you put the chamber down over the soil, it warms the soil, and then you try to find out what happens to the fungi in that soil. So I'll take you through both of these approaches in the Antarctic. So this is the latitudinal approach, and this is where colleagues and I, we sampled soils from the South Orkney Islands, moving all the way down the peninsula, down to a place called Mars Oasis on Alexander Island. I'll tell you a little bit more about Mars Oasis in a few minutes. And there's a difference in mean annual surface temperature at these sites of about, I think it's about 7 degrees C or so. We did this work in collaboration with the Royal Navy. We used helicopters and boats from HMS Endurance, this ship here. And we actually got to some sites that had previously been uncharted, so it was quite it's exploratory science. These are some of the sites. So we always sample what are known as fellfield soils. So we didn't sample soils that had plants on them, because plants have a big effect on fungal diversity in the soil. This is Mars Oasis here. Jenny Island, close to Rotherham. This is a previously unnamed non-attack And then this is actually an exception here. This is Whalers Bay and Deception Island. So there's volcanic activity on this island, but we didn't sample anywhere near the fumaroles. Bethan mentioned about DNA sequencing. So to go into a little bit more detail, what we did in this study was to extract DNA from the soils and to amplify it using a PCR test. This is one of the very few positive things that have come from the pandemic, is I no longer have to explain what a PCR is. It's exactly the same technology as is used to detect SARS-CoV-2 in humans. And then we sequence the DNA using a next generation sequencing method. This was previously something called 454 sequencing, but nowadays it's Illumina sequencing is generally used, and that generates literally millions of sequences in each run. Each of the sequences can be viewed as a chromatogram. And if we zoom in into one little area, you can see all the bases in DNA. So you've got the G's, the A's, the C's, and the T's here in the DNA. And by running these sequences through a publicly accessible database, you can work out which species of fungus you have present in the soil and how many species there are. And that's exactly what we did. We measured all sorts of predictors in the soil. So we measured carbon concentrations, nitrogen, I can't remember everything, but it was pH, water, all the usuals, plus molybdenum even, and various other elements. But the best predictor that we found was actually air temperature. And we found that as the temperature of the sites, or the mean temperature that they experienced each year, increased, this increased the number of fungal species present in the soil. We've got a bit of an outlier here, but generally it's a pretty good increase in the number of species of fungus in the soils with increasing temperature. And we predicted for the southernmost site about a 20 to 27% increase in the number of soil fungus species in that soil by the end of this century. So I'll move on now to the manipulative approach. So this was all done at Mars Oasis on Alexander Island. So if you remember, Rothera is around here on Alexander Island. And if you jump on a twin otter at Rothera, in a couple of hours, you can be at Mars Oasis on Alexander Island, which is a pretty stunning spot. This is the oasis in the foreground, and then this is the Seward Mountains in the background on the Antarctic Peninsula, and then this is all ice shelf here. So this is fresh water, frozen fresh water overlaying seawater. And then in 2007, I was there for a week or so, setting up an experiment using open-top chambers with two colleagues from BAS. We had a lot of fun. BAS, the way it operates and has operated for the last, probably, I'm guessing 70 years or so, has been to take people into the field and for them to stay in camps. So you stay in a tent on the side of the glacier. So the plane lands on the glacier here, and then what happens is the plane lands. The pilot pitches out all the boxes and the tent in a row. He sets up the dipole radio, which is literally, as it says, it's two poles with an aerial between them. He contacts the boat, he makes sure everything is fine, and then he leaves you there with one other person, or potentially one other person, could be two. And this all happens in the space of about 10 minutes. And the first time this happened, I was very, very intimidated because there's literally nobody else on this island. There's two other people that are maybe half an hour's flight away. it would be impossible to reach them on foot. So I kind of got used to it, but it was quite it was quite an experience first time I did it, and it was quite intimidating. This is the inside of a tent. So we're supplied with dried rations. We have a stove here. We're supplied with after, which is aircraft fuel, and matches. And with those things, you can survive because you have literally an almost inexhaustible supply of fresh water just outside the tent. Staying in tents is actually not that uncomfortable. You get to know the person that you're with very well. This is my friend and ex-colleague, Roger, who I spent time with in tents. As I said, we put down an open-top chamber experiment at the Oasis, and what we found is that the chambers increased soil temperature. So the red line here is the temperature within the chambers, and then the blue line is the temperature outside the chambers. You can see here that temperatures are generally higher within the chambers than outside. And we left the chambers there for 5 years, and that in agreement with the longitudinal study that led to higher soil fungal species diversity. So this is inverse Simpson's diversity index here, and these are the samples, or these are the fungal diversity in the samples from within the chambers in red, and then from outside the chambers in blue here. So you can see it's about a doubling in inverse Simpson's diversity index, which is not quite the same as raw species number, as I showed you in the longitudinal study, but it's pretty close. So what we think is going to happen as climate change takes hold in the Antarctic, the diversity of the fungi in the soils will increase. And that is likely because fungi is so important to soil function, such as the turning over of carbon compounds within soils, that will probably alter the way in which the soils work and how efficient they are at processing organic and inorganic compounds. Essentially, they'll become, hopefully, more biologically active soils. So I'm not sure how much time I have left, but I'll just take you to the other end of the earth now, to a place called Newsham on Svalbard. Svalbard's this archipelago of islands off the North Cape of Norway. And this is a research base run by the Norwegians with many different nationalities involved. There's about 14 stations on the Newsham, with 4 being open year-round, and there's about 20 nationalities of scientists visiting this place each year. It's a lovely place to go, and you'll meet all sorts of people at this place. I alluded in the talk outline to encounters with local wildlife. This is the main issue on Svalbard. It's the presence of bears, and it's a little bit disconcerting not to be top of the food chain. They can be quite frequent. It depends on the time of year, but if you go in July, they tend to be raiding bird nests that are near to Newsham. So colleagues and I saw 4 in 10 days this July. This is a young female that we encountered in 2017. We had her full attention, as you can see, but luckily we were in the boat and she was on the shore, so everything was relatively okay. They're beautiful animals. So there have been very significant changes to the climate at Newsham, and in fact on Svalbard as a whole. It's I think supposedly it's one of the fastest warming locations in the Arctic, which I think means, given that the Arctic is one of the fastest warming locations on Earth, that it's most probably one of the fastest warming places on the planet. But there's actual physical evidence for this because people have been going to Newsham for a long time. There are historical photographs showing, for instance, in 1922, the Kronebrain Glacier here. This piece of land here for scale is called Nailoke, and it's 3 kilometers across. And then this is exactly 100 years ago from last summer, and you can see it's receded several kilometers backwards. It's very rapidly receding. And then in 1923, there was a Swiss man called Walter Nistelholzer, who was an aviator. He went to Svalbard and took photographs of Kings Bay here. This is the, I think Newsham is around here. These two glaciers here, this is the Østre Løvembreen, and then the Midtre Løvembreen. This apparently is one of the most studied glaciers on Earth. But you can see here, this is a photograph that I took a few weeks ago when I left Newsham, that this glacier, the Østre Løvembreen, has receded by about 1.3 kilometers in that time. An important ecological process that's apparently caused by Arctic climate change is called shodification. I don't particularly like this word, but I suspect it might have originated over in America. But anyway, over the last few decades, people have noticed the encroachment of shrubby plants has taken place into plant communities that are formerly dominated by mosses. Shrubs are set to become the dominant plant form in the region by the end of this century. All of the Arctic shrubs form symbiosis with soil fungi called ectomycorrhizals. There are some ericoid mycorrhizals. I think Bethan was asked about these previously, but the ones that are really on the increase tend to be ectomycorrhizal plants. I'm sure there's probably a few, actually, looking at this, I imagine there's probably a few ericoid mycorrhizal plant species in here as well, but the majority are ectomycorrhizal plant species. So they form a symbiosis with plant roots, or sorry, they form a symbiosis with basidiomycete, typically fungi, forming these rather lovely structures. So the ends of the roots, they get infected by fungal hyphae, and these the roots, if you like, they swell at the end. They form what's known as a mantle, and these are really important for the uptake of nutrients from soils. So, as Bethan explained, uptake of nitrogen from soils is increased by this symbiosis, and plants in the Arctic strongly nitrogen limited. And I've had an experiment for the last 10 years, close to Newsham, using open-top chambers to examine the role of these symbioses in shrubification. You see here, this treatment rapidly affected the growth of the plants. So this is you see the edge of the chamber here. This is within an open-top chamber in 2014 that we started the experiment. And then this is 4 years later, you see that plants have started popping up all over the place. And this one particular species, Salix pilaris, which is a dwarf willow, is one of the main plant species to have taken hold. There's also another species called Distorted vivipara that has really taken off. And what I'll do next summer is to visit there with a new PhD student and with people from the University of Ghent to try to figure out whether or not these ectomycorrhizal fungi on the roots of these plant species are responsible for the process of shrubification. Okay, so that's the end of the talk. I hope you enjoyed it, and I guess I'm now handing back to Lynne.
Thanks, Kevin. That was awesome as ever. Super.